Hello viewer, welcome to Elimu TV where you watch and learn. Today is the 10th lesson of Biology Form 4 and you're going to learn about the application of genetics in real life. I'm your teacher, Madam Christine Okot. Welcome. This is a preview of what you're going to learn today in this lesson. So first we're going to start by reviewing the previous assignment and the previous lesson. And today we're going to discuss about the application of genetics in real life situations. And lastly, I'm going to give you an assignment. So to start off with the assignment, question number 1 was, explain what you understand by the term sickle cell anemia. And in our previous lesson, we were discussing about the sex-linked gene, and among them was sickle cell anemia. We had hemophilia, which are linked to the X chromosome. Then the sex-linked genes which are linked to the Y chromosomes, we had the hairy nose and the hairy ears. So in our previous lesson, we said that sickle cell anemia is a condition whereby the red blood cells have an abnormal shape. Therefore, they tend to have the shape of a sickle cell. So due to them having that sickle cell shape, this leads to leads to inability of them to transport oxygen. Then explain what you understand by the term a sickle cell trait. So a sickle cell trait is a condition whereby a person acquires both the dominant and the recessive traits, leading to the person being a carrier. So if this person is a carrier, it means that he or she has acquired both the traits, the dominant and the recessive, but it can only show on the phenotype. The phenotype is it just shows on the genotype. The genotype is the genetic makeup. So the genetic makeup will be able to see that the gene is defective, but when you look at the person physically, that is the phenotype. Remember, we described what a phenotype is. We say this is the physical appearance of an organism. So on the outside, you're not going to detect if this person is sickle cell affected, but on the inside, the genetic makeup shows that this person has a defective gene, therefore this person is a carrier. The next assignment, the next question, that is, state the types of gene mutation and explain briefly. So we have 4 types of gene mutation. In our lesson, we discussed all this. And so I hope you remembered all of them. The first one, we have deletion, and you're supposed to explain briefly about the gene mutation. So in deletion, just from the word, it comes from to delete. It means a segment or a portion of the gene has been deleted. Then when you come to the second type of gene mutation, we have that there's a portion which has been included which was initially not there. So something has been inserted in the gene sequence. Then when you come to substitution, this is where a sequence of the gene has been removed and has been replaced with another one which was initially not there. Then when you come to the last type of gene mutation, you have duplication. This is where a portion of a gene has been duplicated or has been have. And to the business of the day, application of genetics. So we say that the knowledge of genetics is very useful in different fields in our everyday to-day life because it helps to improve the quality of life and offer solution to some aspects in life. So after learning about genetics, what next? It's not all about doing exams and filling your brains with genetic knowledge. So we have to apply this genetics in various aspects in our life. For example, these are just a few of the areas where genetics has been applied. The first one is in blood transfusion. The second one is in the artificial selection of plants and breeds. The third one is in genetic counseling, and lastly in genetic engineering. So we're going to tackle each and every of these 4 aspects, and then we see how genetics is used there. So starting off is in blood transfusion, and this is just the transfer of blood from one person to the other. So the person who donates this blood is what we call a donor, and the person who receives this blood is what we call the recipient. So we can just say blood transfusion is the transfer of blood from a donor to the recipient. So successful blood transfusion is based on the patient being given the right type of blood from a donor. So it's not every recipient can receive any type of blood. So this is done by something we call blood typing, which is based on the genetic knowledge of the blood group. So we have 4 blood groups. We have O, A, B, and AB. Basing on the antigen which is found on the type of blood. So if you are an O person, it means that you can only donate your blood to a recipient who has O, has A, B, and AB. So we say O is a universal donor. He can donate to all blood groups. Then when we come to B, to A can only donate blood to somebody who has a recipient who has blood group A and blood group AB. Then when you come to B, a donor who is B can only donate blood to a recipient who has blood group B and AB. And finally, if you are blood group AB, you can donate to somebody who has AB. So this one is based on the genetic knowledge of the gene, the makeup of the blood types. So not anyone can donate blood to any person. So it has to be the specific type, and also you have to consider the rhesus factor. If you are an O negative, you cannot donate to an O positive. So you have to have the same blood type and the same rhesus factors. Then another area been applied is in the artificial selection of plants and in breeds. So this basically involves the choosing of individuals that have certain desired characteristics and crossing them. If you have looked at a certain animal or a certain breed of plant, and you desire those characteristics that animal or a plant has, so you simply just cross it over so that the next offspring can give you the qualities that you desire. So the main purpose of artificial selection is to help in the qualities such as fast growth. You will need the plants or animals that will grow at a higher rate. Then in disease resistant, you need crops or breeds that are resistant to disease. They are also resistant to the pest, and they can tolerate all type of climate. For example, if it's the hot and extreme climate. Then if you have all these qualities, it helps you to yield and increase production in your food. And if you have a good production in food, this leads to food security. For example, if you have a bull and you have noticed that it has the genetic makeup of something which is resistant to a disease, then you want to cross it with a cow which is producing high volumes of milk. So what you just do is you do the crossing. So you have a bull which is homozygous dominant. It means that the traits are very dominant. Then you cross it with a homozygous dominant cow that is high in production of milk. The offspring that you're going to get is going to be a codominant allele. It's going to take all the genes which are dominant, and your offspring is going to have double traits. That is, it's going to be resistant to diseases, and it's also going to have high production of milk. So that is an aspect where artificial selection of breeds is applied. Now moving to the plants, still the same aspect of artificial selection. The plant breeders use a chemical that is called colchicine to develop what we call polyploidy. And in our previous lesson, we discussed about polyploidy. And just a brief reminder, we say that this is a chromosome which causes the formation of multiple number of the chromosome. Initially, the chromosomes are supposed to be diploid. So this means that they're supposed to have 2 sets of chromosomes, one from each parent. So one parent has N and the other parent has N. So it's supposed to form a diploid. That means the cell is supposed to have 2N. So what happens? This is the occurrence, we've just described the polyploidy, is the occurrence of additional chromosome due to chromosomal mutation. So this chemical we call colchicine is applied to the terminal buds of the branches. So the cells are tetraploid. So initially they were 2N, now they are 4N. It means that it has double up. So the size of the fruit or the size of the plant is also going to double up, and it's going to be larger than the normal fruit. So these are just examples of where polyploidy has been applied when you take that chemical colchicine and apply it. So for example, there we have the orange.
if you can look at the normal size of the orange is smaller than the other one whereby polyploidy has been applied, so it's going to produce a fruit which is larger than the normal size. The same applies to that strawberry that you see there, because you know the normal size of strawberry is smaller than that. So when polyploidy has been applied, it means the size is going to increase. The same applies to the illustration of the watermelon that we have in that area. The next aspect or the next area where genetics is applied, this is in the genetic counseling. So this is especially for couples who have hereditary diseases. So they need this counseling so that they can know about the probability of their offspring inheriting the disease, some of the preventive health cares that they're supposed to give to their children who are affected, and also to see if there are possible preventive measure. For example, there are some couples who might have inherited the diseases, maybe they were carriers and they did not know. So if they give birth to a child who is affected and they have never ever experienced how to bring a child who is affected with any type of genetic disease, they need counseling so that they know how to give this child the best medical attention and what to do so that to prolong the life of their child. So those are some of the areas where people, the couples, need to go for genetic counseling. Then also it helps to decide disputes on parentage. So in this case, we use the ABO system of paternal dispute, and that involves the test on the DNA. So the issue of I'm not the dad, I'm not sure how happened. So there's a way in which all this can be solved. So you just go and do the DNA using the ABO system, and the dispute is settled using that. So genetics is very useful in that field. Then another area, this is a process where useful genes are transferred from one organisms to another by human manipulation. So the fourth application of genetics is in genetic engineering. So you take the useful genes and transfer them from one organism to another so that you can get the qualities that you desire. So this is mainly done by altering or changing the DNA molecule to increase the crop yield. It can also be helped to improve the quality of a certain organism, and it can also be used to prevent the genetic disorder in the offspring. So genetic engineering has been applied in two major fields. One is in the field of medicine and the other one is in the field of agriculture. So we're going to look at all these areas. So first, starting off in the field of medicine, it is applied in controlling some diseases. For example, the diabetes mellitus. So it is used in the production of insulin by using a bacteria. So what happens here, following our illustration here, there's a transfer of gene that determines the insulin production in human, and it is inserted into a bacteria. Why bacteria? Because bacteria multiply at a faster rate. So when it has been applied there and the bacteria has multiplied, the insulin now is extracted from the bacteria and it's purified and ready to be used by the patients who are diabetes mellitus. Now, in the field of agriculture, genetic engineering has been applied in the following area. For example, you want to improve the shelf life of your tomatoes. So you use this aspect of genetics, you just insert the gene that will help the tomatoes not to go bad easily. As we know, the tomatoes, after they have been ripe, they tend to go bad very easily. But now when you use these, when you apply the genes which help the tomatoes to have a longer shelf life, so this one is a plus to the farmers. And talking about the genetically modified foods, the GMO, I know all of us have encountered such type of food, the GMO. So you just look at the desired qualities that you want in your plants and you insert the gene in the specific plant that you want. For example, if I wanted my peas not to have the pods, which are green, normally green, I just insert the type of gene that I want. For example, in this case, orange. So when these peas, they grow, they mature up, they will form these pods which are green, orange in color rather than the usual green. Then if I desire the apples maybe to have a touch of the watermelon, so just insert the gene, then you'll have something like that. So it's an apple and it has some aspect of watermelon inside it. Then if you desire your fruit, let's say an orange, to have a variety of flavors or a variety of things, for example, you want it's an orange but it also have the kiwi, so you just insert the gene of the kiwi fruit and it will develop. You want maybe to alter the color of the fruit, the orange, because we know orange is orange in color. Now if you want to alter the color of it, you just insert the correct gene in the best proportions. Then we have this amazing food which has been genetically modified. For example, you want your bananas to have some aspects of the kiwi fruit. So on the outside it looks like a banana, but on the inside it's that fruit of a kiwi. So these are some of the items or the food which have been manipulated using genetics. Then it also helps in to make the crops to be resistant to the pest. So we know a lot of crops, they are affected by the pest. So what happens is a gene can be transferred from a bacteria, that bacteria is called Bacillus thuringiensis, into some of the crop. So when it is inserted, the gene triggers the production of substance that will repel the pest. So when these pests come to devour your crops, they will die because of this substance that repels the pest, and your crops will not be affected by any type of pest. So this one is a plus to the farmers and something very good from the genetics which we've learned. So to mark the end of our lesson, I'll leave you with this assignment. Question number 1, what is polyploidy? Question number 2, how do plant breeders use polyploidy in plant breeding? And the last question, give a reason why the study of genetics is important during blood transfusion.
I hope you've enjoyed today's lesson and learned something. For any questions, feedback or clarification, you can hit our Facebook page, Elimu TV. Our Twitter handle is @Elimu_KE. Until next time, I've been your teacher, Madam Christine Okot. Keep it Elimu TV, where you watch and learn.